Hello class, my name is Hassan Kurma and today I'm going to talk about Enzyme, which is our project. But first, this is our audience right here, Roxy, which she's going to give me energy to talk about Enzyme a little bit. Enzyme is a substance produced by living organisms that acts as a catalyst to bring about the specific amount of biochemical reaction. It's six substrates and their shape must match the active site to make product. And its lower kinetic energy as needed, and heat can be denatured. The saliva contains amylase and digests starch into maltose, and the stomach produces the pepsin and it digests the protein to amino acids. Pancreas inject the enzyme to complete the digestion, and large carbohydrates are digested into glucose. And that's a little bit of information that I got about enzyme. And these are some of the products that we're going to use today to talk about enzyme and make the cupcake so you guys can enjoy it. Thank you.
Class, today we are talking about the topic enzymes. There are different parts to the process that helps us understand better. Right now we are going to focus on metabolism. Metabolism refers to any chemical process occurring within or between the cells. Most chemical reactions within the cell do not occur spontaneously. Spontaneously means not by magic, not fast. In metabolism, it is a catalyst. The enzyme is the catalyst. Enzymes bind with different kinds of substrates. For example, we have here today, we have water, which is part of it. There's also eggs. There's cake mix, and because of that, it's because there's sugar inside. And then we have the actual enzyme for today is this bowl. This bowl represents the enzyme itself. Until this chemical reaction occurs, they free themselves, and because of that, the finished product is this chemical reaction is the cupcake. The enzymes are not changed by this chemical reaction.
Hello class, my name's Jovi, and today we're going to be talking about the chemical reactions found in enzymes.
Hey!
Hey Karina, how are you?
Good, how are you?
Awesome.
So are you making cupcakes today?
Yeah, I am.
Can I join you?
Definitely, Karina.
Okay, can you tell me about the process of different enzymes and how to make them?
Sure. So, in enzymes, there's different parts, items, and processes. The first part in the chemical reaction is we need our active site because it has a small port for the enzyme. So here are examples of active enzyme. We have a big one, the bowl. We have a big active site and small active sites. So there's two different sizes. And then, Karina,
what happens after that?
after we have those, we're going to need the substrates. The substrates are the molecules that go into the active site. So it would be the eggs, the cake mix, water. They go straight in here into the small little port.
So what happens after that?
After this, when we mix our ingredients and put them into our active site, they go through the lock and key process. Here, the ingredients or the molecules bind to the active site.
Do they stick? Is that what you mean?
Yes, they do stick. And
so they stick. Does that mean that they go like this and they just stick to the bowl?
Yes, Karina, exactly what you just did.
So what happens after that?
After that, we put our ingredients or the substrates in the oven and we let them bake. From there, once they're done, we take our products out of the active sites, and they leave the place that they were, which is the active sites.
Oh, cool. That's interesting. Let's get baking.
Awesome. Let's do it.
Hi, good afternoon. I'm Sean, and today I'm going to be just discussing with the class briefly about cell inhibitors, what they are and what they do. Okay, so there are basically two... Oh! Hassan, is that you?
Hi.
Hi, you scared the crap out of me with that horse mask. You scare me more every year, and I gotta be ready for it so I don't get a heart attack.
Okay. Can you teach me a lot about inhibitors?
Sure. Alright, well, an inhibitor, there are two types of inhibitors. In a sense, there are the human-made inhibitors, and then there are the naturally occurring inhibitors inside the human body. So, the naturally occurring inhibitors are polymers which inhibit or prevent your body's cells from carrying out whatever process they try to carry out. And there are two types of these naturally occurring inhibitors. One is a competitive inhibitor, and the type that's called a competitive inhibitor will park itself right at an enzyme, at an enzyme's active site, and just completely block off the substrate that's trying to park itself there too. Then there's the shape-changing inhibitor, which you could argue is a lot more evil because it latches on to the enzyme's active site and then it completely deforms the shape. So when the substrate that wanted to park there comes by, it can't because it realizes that the active site has changed shape completely, along with the enzyme, and it can only get back in there if the enzyme were somehow to revert back to its original shape. And then there are also the human-made cellular inhibitors. One very nasty example is related to mustard, but a lot more lethal, deadly, and just negative for humans. Mustard gas, not mustard, mind you. Mustard gas tastes horrible and takes lives. Was used in World War I by the Axis powers, and basically it can do a terrible thing to your nerve cells, which is stop them from functioning, which you need for the rest of your body to function. And after World War II, there was a Geneva Convention on proper international etiquette in war, and they determined that using chemical warfare like mustard gas should be illegal, and so it is illegal to this day to use that in war. So, Hassan, I hope I've given you a better idea of what inhibitors are and what they do.
Thank you so much.
Yeah, you're welcome. Thank you for being an audience.
Please science class and learn a lot about science. Peace.
Hey, everybody.
Hi.
Hello, what's up?
Hi.
So now we've got a whole bunch of items used to bake cupcakes here, and we're going to use them as analogies to demonstrate to everybody how certain cellular processes work.
And how certain items work too.
Like, for example, as Sean said, he talked about inhibitors. So we are now going to show you different examples of inhibitors, like the examples that you have talked about. So
We hope you guys enjoyed the video.
we hope you enjoyed the video and enjoy spending time with us here today. So it's great. So,
Thank you.
We'll see you.
Peace.
Peace. And hopefully you guys are having a good time watching this.
We did all the batter and everything.
It
put
looks
all together.
awesome.
It's perfect.
I bet these cupcakes are gonna come out awesome. We're gonna eat them tomorrow too.
I know, exactly.
Oh man, this is gonna be great.
Excuse me, I noticed you two were baking here, and I'm a nosy person, so I don't really approve of your cooking, and I'm gonna show it.
Hey, Sean, you know what?
Hey, what the? Oh, it's an antidote. You know what? These cupcakes don't look that good anyway, so you know what?
Get away! These are my cupcakes.
Oh, what do you think this is doing right now? Because I put this here. Why do you think I put it here?
Well, I think you put it there, Karina, because it's gonna bring you to the next topic, which is denaturization.
Remind me what that means.
Alright, Karina. Denaturization is when the ingredients in the active site, they end up going to changes in temperature and pH. So as a result of that, they end up switching their shape over time.
Oh, cool. That sounds really interesting.
For example, since we left these here, when we go to bake, they're going to go through that process as the mix rises up.
All right. That sounds like a really interesting thought. So,
Well, anything else you'd like to know, Karina?
let's get baking.
All right, let's do it.
So, we worked really hard today just trying to explain and show you guys, demonstrating how enzymes work and the different variation of the process. So, what we have here today, right?
Yes.
We finally did the
cupcakes. Yes, that we've been using to demonstrate.
Right.
Yep, and it's right here. In fact,
Look at the results.
Yeah, the results of that, and special credit to Karina's mom, Alice, for baking them in the oven so accurately after we, made
made the batter.
batter. Yeah.
And I would like to say thank you for you guys for being willing to come over, and we worked so hard on this video, and it was so much fun. I had a hilarious time.
And I had an awesome
What about
time
you?
too, Karina.
Oh
And
yeah,
you too,
I
Sean.
had a great time. Also, special thanks to Karina's entire family for letting us use their kitchen and other spaces to make this video possible.
And I like to thank Hassan for videotaping a lot.
You're welcome.
And for
all
joining
of
in
that
too.
as well.
Thank you.
And we've been working really hard, so hope you guys enjoyed the video.
And hope you
I
guys had some
a great
awesome
time
laughs.
laughing hysterically to all mishaps and stuff. So, thank you.
You're
Bye.
welcome.
Enjoy.
Hello guys, I'm here today to teach Sean how to bake a cupcake. So Sean,
Yep.
these are the stuff here I'm going to teach you about how to make a cupcake. So first you can take all this out and transfer this on the other bowl,
Okay.
and you're going to mix it when I'm done.
Okay.
Right? So there we go. So now you can
All right,
mix it
time
up.
to mix. All right.
So you can use that
Okay.
and mix it and see how it's going to look like.
Okay.
Put it on medium.
Okay. Medium.
Let me help you.
There you go, right there.
And it, okay. Oh, and it says to do it for 2 minutes.
2 minutes. Okay.
Be careful, you don't want to hurt yourself.
True.
Use the spatula while you're doing it to make sure that he doesn't get any crumbs.
Make sure there are no crumbs left.
Okay.
I think it's been more than 2 minutes.
Okay. Should stop now then.
Yeah, I think that's good. You can just put it right here.
Okay.
There you go. And now we're gonna transfer the cake on the little pans so they can bake.
Why don't we just... That's so good that you cracked it like perfect. Oh wait, there's a shell. Oh. You know that movie I really want to see so bad?
Which one, Karina?
Cinderella. I
No
mean,
way.
seriously.
Oh my gosh. And the one I've been dying wanting to see is Insurgent.
Hello
That's-
everybody, I'm Erwin, the irritating inhibitor, and I see these two ladies here trying to demonstrate cellular processes and bake cupcakes, and well, they're not doing it to my liking, so I'm going to be a pain and try to inhibit them from baking.
Hey,
Oh, sorry. I was leaving anyway. You know, those cupcakes didn't look that good anyway. I think he pushed me a little bit early. Out a little
Wait,
bit
what's
early.
going on here?
Because I was trying to well, I think.
No, what?
No.
You're not supposed to tell.
Good afternoon, class. I'm Sean, and today I'm just going to discuss with you all briefly what a cellular inhibitor is and what it does specifically. Hey, it's you, Hassan. Hey!
How you doing, Sean?
I'm okay. This is an awesome St. Patrick's Day costume. I swear you outdo yourself every single year.
Thank you so
Thank
much
you,
for
Hassan.
letting me learn a lot about inhibitors. Students, please stay in class and learn a lot about inhibitors. Peace.
Hello class, my name is Hassan Kurma. But first, before I start, I want to talk about Enzyme, which is our project.